I had started the introduction on the organs of the immune system, and I had described about the positions or regions in where these lymphoid organs are or immune system organs are situated. Okay. Today we will first take up the primary lymphoid organ, that is thymus. I have told you that it is in the chest region and it is almost on our systemic trunk, and it is a two-lobed structure. And when you take the section of that and histological preparation, when you prepare, it will look like this. You can magnify this further under the microscope, but this is the low magnification photograph just to show you how it is zonated. So the outermost covering of the thymus is called as capsule. It is made up of connective tissue. And this capsule connective tissue runs inside with projections inside, which is called as trabeculae. Trabeculae. I will show you a diagrammatic presentation in the next slide. But just remember the indentation or inward projection of the connective tissue capsule makes the thymus to divide itself into chambers, and those chambers are called as thymic lobules. Okay, and we'll see how it looks in the diagrammatic presentation. Here it is labeled as cortex and medulla, means this lobule, that is the lobule that is surrounded by the connective tissue here, having a central area called as medulla, and the peripheral area is called as cortex. So if you take one of the lobule, and that one lobule can be picturized diagrammatically like this. The capsule made up of connective tissue, which is indented as trabeculae, and in the trabeculae the blood vessels are there, that is both the arteries and the veins, which are responsible for draining the thymus as well as supplying the blood to the thymus, are present there in the trabeculae. And the internal matrix of the thymus, is made up of the stroma, the stromal cells, which are mesenchymal cells, and plus other cells which are there. And most important cells that are there in large numbers are thymocytes, and thymocytes are nothing but T cells. Okay, thymocytes are nothing but T cells. And from where these thymocytes originate? They originate in the bone marrow as progenitor T cells, and they have a receptor on their surface which is called as homing receptor. And that homing receptor which is there on their surface, which is responsible for them to be brought to the thymus. They are released from the bone marrow in the blood because of the homing receptors through the blood as well as from the lymphatic channels. The lymphatic channels which come into the thymus bring these progenitor T lymphocytes, which are called as thymocytes, and they come and lodge themselves in the thymus. So the area which is labeled as cortex, that is the area which is next to the capsule, next to the capsule, consists of large population of incoming thymocytes, means young, non-committed, or the T lymphocytes which are not stimulated for any immune response, which can be called as naive, Naive, naive T cells. Naive T cells means they are not yet committed to any antigen. Newly arrived T lymphocytes will be there in the cortex area, and they'll be in large numbers, and they are going to be nourished by special type of cells are there called as nurse cells. And sometimes these thymocytes are enclosed within the nurse cells, and these nurse cells provide nutrition to the thymocytes. You can see here in the cortex area, some nurse cells are surrounding it, and some nurse cells have within them the thymocytes. And by nourishing those thymocytes, they make these thymocytes to undergo proliferation, means mitosis. And these pro T cells or progenitor T cells undergo mitosis in the cortex area to form a large population. Then the nerve cells which nourish them, which are modified epithelial cells. Then comes the other type of cells, which are called as simple thymic epithelial cells. And the thymic epithelial cells can be in this cortex region as well as in the medulla region. And the cortical epithelial cells, which are there, are capable of the selecting the thymocytes to commit themselves to become TH cells and TC cells. TC means cytotoxic T cells. TH means T helper cells. Okay, so the process of that selection, we'll talk about it when we are going to talk about the T cell maturation process. For the timing today, we'll just learn the function of the thymus for selection of T lymphocytes to commit themselves to become TH and TC cells is done by the cortical thymic epithelial cells. Then there are other cells which are of the immune cell type. One of them will be in fact macrophage, and the macrophage which is there is also responsible for the selection of the T lymphocytes. And there are interdigitating, which I told you, like finger-like projection, interdigitating dendritic cells are also there. It is labeled here interdigitating dendritic cells. So interdigitating dendritic cells and the macrophage which are present, and they are in large numbers in the medulla region compared to cortex region, and they help in negative selection of the thymocytes or T lymphocytes. Positive selection of the T lymphocytes is done by thymic epithelial cells, and negative selection of the thymocytes or T lymphocytes is done by the antigen presenting cells, the professional antigen presenting cells, that is macrophage and interdigitating dendritic cells. So the cells, the T lymphocytes or thymocytes, as they go through the maturation process, they descend from cortex area to the medulla area. And these thymocytes can leave the thymus and go to the lymphoid organs, other lymphoid organs. Other lymphoid organs could be even bone marrow, they can go to bone marrow back, or they can go to lymph nodes, they can go to spleen and adenoids and tonsils, etc. Visit that place and come back. Okay, and they keep doing that. They leave thymus, come back, leave thymus and come back, and this trip which they undertake is 48 hours cycle. And within 48 hours cycle, if they do not come across any antigen of the foreign origin, they will commit suicide, that is apoptosis. Okay, so the thymocytes which are proliferating in the cortex region undergo selection in the cortex region by positive selection, and in the medulla region by negative selection using the APCs, and they leave the thymus and go to the secondary lymphoid organ. And if they come in contact with the foreign antigen in the secondary lymphoid organ, they will never come back to the thymus. But if they don't come across any foreign antigen, they will come back to the thymus. And next, once again, they will leave the thymus, and that cycle will be repeated. But there is some duration, like most of the thymocytes last only for a week. Before that, if they have not come in contact with any foreign antigen, they do not last beyond 1 week. So please remember, thymus is a primary lymphoid organ, and it is very important for the selection of TH cells, that is T helper cells, and TC cells. And the T helper cells are the most important cells responsible for the next arm of the immune response, and that is the humoral immune response. Antibody production by the thymus, antibody production by the B lymphocytes is dependent on the T helper cells. And without the T helper cells, the antibody production cannot take place. And if at all it takes place independent of the T lymphocyte help, then it will not be long lasting immune response. Okay. Then we'll take up the another lymphoid organ, that is secondary lymphoid organ, spleen. The spleen in histology looks like that. In that you can see some blue patches, island-like patches, which are being labeled as white pulp. Blue patches in the histology, they are white pulp. Then a nice powdery granular mass which is dispersed uniformly, and that is called as red pulp. So red pulp is the area where RBCs are in large numbers and scanty B and T cells. B and T cells are scanty there. Similarly, very less number of the antigen presenting cells will be seen in that area, because spleen is a major organ, that is a secondary lymphoid organ, which is responsible for building an immune response for all those antigens that arise from the gastrointestinal tract. Any antigen that is coming through the gastrointestinal tract is communicated to the immediate lymphoid organ, that is spleen. And spleen is the first organ to respond to all those antigens that enter the gastrointestinal tract. And those RBCs which are produced fresh, that is from the bone marrow, which arrive as proerythrocytes, undergo maturation in the spleen and get distributed to other parts of the body through the spleen as non-nucleated RBCs. RBCs, when they originate, they are nucleated, and those are proerythrocytes. And some of the proerythrocytes can come from bone marrow and lodge themselves in the spleen, and then finally transform themselves into the RBC, anucleated, means without nucleus, by expelling the nucleus, and from there they are distributed to other parts of the body. Another important function of the spleen is those RBCs which finish their life cycle of 120 days. You know that RBCs have a lifespan of 120 days. And if they finish their lifespan of 120 days, those RBCs are going to be destroyed in this area also, that is in spleen. And the second major organ where the RBCs are going to be destroyed is the liver. Liver and the spleen are the two organs where the final destiny of the RBC is decided. Those RBCs which finish their life cycle, they are undergoing processing here, destruction here. Okay, and that all happens in the red pulp area. Red pulp area is majorly dedicated to the RBC. Therefore, in histology also it looks almost reddish in color because of abundance of the RBC. Then comes these island-like areas which are called as white pulp. And what is this white pulp? The white pulp in diagrammatic representation, we can take it up like this. The white pulp area is nothing but, you can make out here the congregated large number of blue cells here. Large number of blue cells. And that large number of blue cells is forming an island. And what is that blue staining cells? They are nothing but large population of T lymphocytes and the B lymphocytes congregated in one area. You can see such islands in large numbers. You are seeing those islands in large numbers in the histology. That is indication that they are called as lymphoid follicles. Okay, lymphoid follicles. The white pulp, it is labeled here on the right hand side of the diagram as white pulp, and that is what they are explaining. The white pulp in transverse section, it looks like an island with large number of population of B and T lymphocytes. In the central region of that island, You can have blood vessels, and that blood vessel is shown here. Okay, the blood vessel, that is artery, and the veins could be there. All right. Majorly, it is supplied with the arteries, and that artery is called as the major artery that is responsible for supplying of the oxygen to those lymphoid cells which are surrounding it. Now, that artery is surrounded by, you can see the blue dots around that artery. Okay, blue dots, and those blue dots correspond to the lymphocytes. In inner part, that is immediately close to the artery, are the T lymphocytes, and in the periphery of them is the B lymphocyte. And that area which is surrounding that artery with large population of lymphocyte in cross section looks like this island or the white pulp. And what is that area is called as? It is called as PALS, P-A-L-S. PALS stands for periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath. It is nothing but the large population of lymphocytes surrounding the artery. Okay, and these lymphocytes which are there waiting to be activated by the presence of antigen, and that antigen is going to be communicated to them or presented to them with the help of the antigen presenting cells. And those antigen presenting cells, I told you, they are interdigitating dendritic cells which will be there in the central area here, as well as the macrophages. Those are the professional antigen presenting cells. In addition, you would see in this follicular area follicular dendritic cells. I told you about that. And those follicular dendritic cells will be also responsible for the activation of B lymphocytes. Okay, and the area initially that it may not contain such a large population, but when they come in contact with the foreign antigen, it undergoes enlargement, and that would result in swelling of the spleen. When the spleen swells, you can palpitate it. You can palpitate it by pressing the area in the abdomen just below your stomach. Sometimes when you have complained of stomach ache or intestinal pain, internal intestinal pain in the intestinal area, abdominal pain, okay? Abdominal contains the intestine, stomach is in the upper region. You can have stomach pain, you can feel that it is in the stomach. If it is paining in the intestine, you can feel that it's in the abdomen. Doctor will press that area and palpitate it and see whether there is swelling of these two areas. One. Second, he will palpitate below the stomach, in that left hand side of your abdomen and feel the swelling of spleen. If he can feel it on his fingers by pressing the swollen spleen, it is an indication that you have GI tract infection. I told you that any infection that has taken place in the GI tract and any antigens that you have come across in your intestine are communicated to the immediate lymphoid organ, that is your spleen. And spleen will undergo massive activation, and those lymphoid follicles which are inactive in the beginning are called as primary follicles. Okay, means they are not yet committed to the antigen, but they are having the B lymphocytes and T lymphocytes which are naive, means new, which are there. But as soon as the antigen is encountered by the dendritic cells and the macrophages, they process it, they will start presenting it to these T and B lymphocytes. When they start presenting it, they will undergo massive activation and proliferation that results in swelling of the spleen. And with the result, the primary follicle will get transformed into secondary follicle, and that secondary follicle also named as germinal center. Germinal center. Why it is called a germinal center? Because there is tremendous amount of proliferation activity in that area whenever the foreign antigen is encountered. Okay, like thymus, spleen also is covered by a capsule, connective tissue capsule, and that capsule is indented into the cortex area and extending up to medulla area to form the zones of the spleen or splenic lobules. Okay, splenic lobules. And those indented areas of the capsule are called as trabeculae. I've already mentioned that in the thymus. Okay, the trabeculae compartmentalizes the spleen, zonates it into different compartments, and spleen having red pulp, which is shown here with the red color, and white pulp, which is shown with large number of blue cells, there is the major area as a lymphoid organ. And white pulp is the area where T and B lymphocytes undergo commitment and activation against a particular antigen. And the periarteriolar sheath, which is present there, lymphoid sheath, pars, is the naive B and T lymphocytes which are present there. You can see that the spleen is drained by a vein and it is supplied by an artery. And periarteriolar sheath is present around the artery, and vein system doesn't have the support of the B and T lymphocytes. Okay, spleen is also drained by the lymphatic vessels, and those lymphatic vessels can make these activated T and B lymphocytes to travel from spleen to another lymphoid organ, or they can help carrying of the antigen from the spleen to other lymphoid organs to increase the immune response. So that is as far as spleen is concerned, which is a secondary lymphoid organ. Now we will go to the another secondary lymphoid organ, that is the lymph node. Lymph node, once again, look at this, it almost has the resemblance to the spleen, and there are large number of island-like structures, and these island-like structures are called as germinal centers, and nothing but they are lymphoid follicles. Lymphoid follicles. And those lymphoid follicles which are light patch in the center, they are secondary lymphoid follicles or the germinal centers that have undergone proliferation, and they are committed to a particular antigen. That is why they have become light area in the middle, that is called as light zone, and the dark zone area around the periphery of those islands. But the fresh or newly formed lymphoid follicles in the lymph node, where the naive T and B lymphocytes are present, or only the naive B lymphocytes are present, and those areas will be dark staining patches, and they are called as primary lymphoid follicles. A lymph node is almost elliptical in shape, or it could assume a kidney shape, like this. It assumes a kidney shape like this, with a large lymphatic channel draining it, large lymphatic channel draining it, which is called as efferent lymphatic vessel. Efferent, EFFERENT, efferent. Okay, efferent lymphatic vessel drains the lymph node, means anything that is present in the lymph node leaves the lymph node from that vessel. Then what are the vessels that come and join the lymph node? And those vessels are afferent, AFFE. Please note the spelling. Afferent, AFFERENT, are incoming. Afferent lymphatic vessels or lymphatic channels are incoming. Efferent are outgoing, and efferent is only one, but afferent are many. I told you that large area of the tissue spaces and body spaces are covered by these afferent lymphatic channels, and they drain the different regions of your body and bring the lymph from different regions of the body and empty it into the lymph node. And when they empty into the lymph node, they can empty the pathogen or the antigen concerned with the pathogen into the lymph node. Therefore, the secondary lymphoid organ as lymph node can become the center of activation of both Th cells as well as B cells. All right. And Th cells which leave the thymus come and lodge themselves in the lymph node in the paracortex area. Paracortex area is the mid-zonal area. This middle part of the lymph node, mid-zonal area, is called as paracortex. And the cortex area is the peripheral area. Peripheral area contains large number of lymphoid follicles, and those are the primary lymphoid follicles. Primary means they have not yet committed to the antigen. Primary lymphoid follicles will be there on the periphery, and they are rich with B lymphocytes. Okay, rich with B lymphocytes in the periphery. Then almost at the border, it is made up of connective tissue capsule, which may be also indented inside like trabeculae, zonating the lymph node into different chambers. And the middle area of the lymph node is called as medulla. And the medulla area of the lymph node is sparsely populated, but it contains all committed B lymphocytes. The B lymphocytes that had committed to the antigen in the paracortex area will migrate from there after activation and come to the medulla area and differentiate themselves into plasma cells. And after they differentiate into plasma cells, they are committed to produce the antibody against that particular antigen, that is foreign antigen, which they had come across in the paracortex area. And with the help of Th cells, T helper cells, they have undergone activation in the paracortex area. So that means in the paracortex area, the B cell activation with the T lymphocytes, that is T cell dependent B cell activation, takes place in the paracortex area. At the same time, in the paracortex area, the Th cells are also committed to the antigen, and that antigen has been presented to them by the professional antigen presenting cells, that is follicular dendritic cells, as well as the interdigitating dendritic cells and the macrophages. Okay, follicular dendritic cells majorly provide antigens to the B lymphocytes. The interdigitating dendritic cells and the macrophages present the antigen to the Ts cells. Now look here, the primary lymphoid follicle has been shown with a light area which is not yet committed to the antigen, whereas the germinal centers that are shown in the label having in the center the antigen presenting cells surrounded by the B and T lymphocytes, and the germinal centers are also called as secondary lymphoid follicles. As I had mentioned in the spleen, the secondary lymphoid follicles are the ones which are showing excessive proliferative activity, and large number of the T and B lymphocytes are committing themselves to the antigen that is being presented by the antigen presenting cells in the secondary lymphoid follicle. So that means the migration of cells takes place. Initially, the cells arrive into the lymph node through the afferent lymphatic vessel. Whether it is a B lymphocyte or whether it is a T lymphocyte, both of them which are naive means new, not committed, arrive here into the cortex area through the afferent lymphatic vessels. Okay, and the B lymphocyte originate in the bone marrow. Okay, B lymphocytes originate in the bone marrow. T lymphocytes of course originate in the bone marrow, but they actually undergo maturation process in the thymus, and after the maturation process, that is positive selection and negative selection, those which leave the thymus, they arrive into the lymph node through the afferent lymphatic vessel. After they arrive here, they are congregated as primary lymphoid follicle. But T lymphocytes try to migrate from that primary lymphoid follicle to the paracortex area and wait there for the activation. And in the primary lymphoid follicle, the B lymphocytes which are there, which have arrived from the bone marrow, where after the maturation process that has taken place in the bone marrow, which have now committed themselves to develop into antibody producing cells, they have arrived here. And once these cells arrive here, they show limited proliferation in the beginning. But as soon as the antigen presenting cells encounter a foreign antigen that is arriving through the afferent lymphatic vessel, those antigen presenting cells are activated. They will release the cytokines and they will warn the T lymphocytes, especially the IL-1 released by the macrophage wants the T lymphocytes which have arrived here to start getting prepared for getting activated. At the same time, had mentioned that the IL-6 which is produced by the macrophage wants the B lymphocyte that there is a foreign antigen. So like that, both these cells have been given a stimulus by the antigen presenting cell, especially the macrophage, which is a cytokine producing cell. Once they have sent these messages, these lymphocytes will not keep quiet. They will now start getting activated. What is activation? Nothing but undergoing proliferation. And once they proliferate, the colony of these cells starts increasing. And since the limited space which is there, that makes them migrate to the central area, that is paracortex area. As they migrate to the paracortex area, before they migrate, they will have been presented with the antigen by these antigen presenting cells. And remember, B lymphocytes can also act as non-professional antigen presenting cells. They themselves have the ability to encounter the antigen on their own, other than the antigen being presented by the antigen professional antigen presenting cells. If they encounter the antigen on their own, they are capable of processing that antigen and then present that antigen on class 2 MHC. And by that time, they will have migrated to the paracortex area, and that class 2 MHC, which is carrying that foreign antigen processed by the B lymphocytes, will help in establishing the contact with the TH cell, which has come in the paracortex area. And now both these cells will undergo simultaneous activation. And the association of TH cell and the B cell, it forms a complex, and that is called as TH and B cell synapse. They contact with each other through receptors. The TCR receptor of the T cell contacts the class 2 MHC, which is presenting the antigen, and a synapse is established. Once that synapse is established, both these cells undergo activation, and that process takes place in the paracortex area. And the paracortex area is considered to be light zone. Okay, light zone means there are cells which are highly distributed and they are in fact well dispersed, whereas the cortex area is called as dark zone because it stains darker. And in this picture you can make out the cortex area is the dark zone and the medulla area is the light zone, and the paracortex area, that is immediately after the cortex area, is also called as light zone. So light zone area is meant for the activated TH cells and the B cells. whereas the cortex area, which is the dark zone, is meant for naive T cells and B cells to arrive and remain there for short duration. Okay. So the activated B lymphocytes, because of the synapse with the T lymphocyte, are the ones which are committed now for lifelong. And these are the B lymphocytes which will remain in your memory, immune memory, and have committed to a particular antigen. Okay, remember when I had dealt with antigens as far as coronavirus or salmonella bacteria, I had mentioned that a given organism can have many antigenic determinants or epitopes, right? We had classified, remember, the spike protein, then the membrane protein, nuclear capsid, envelope protein, so many things are there. All of them are different ones. And each B lymphocyte can commit for each one of them. Similarly, for H antigen of salmonella, one B lymphocyte will commit. VI antigen, another B lymphocyte will commit. Then O antigen, another B lymphocyte will commit. Like that, three different B lymphocytes committed to three different antigens will be responsible for independent production of three different antibodies. So like that, many epitopes, many antigenic determinants will be responsible for different B lymphocyte activation. And each B lymphocyte that is activated, if it establishes the synapse with the TH cell, that B lymphocyte is committed to that antigen for lifelong. And it will remain in the memory of the bone marrow for lifelong. And that TH cell, which had committed to the antigen that was presented on class 2 MHC, will remain as T memory cell lifelong in an individual. So the process of acquired immune response, acquired immune memory, all that will begin in the lymph node. Of course, there are plenty of lymph nodes. You know that I had shown you in the previous diagram. There is a huge channel, like there is a central channel of lymph nodes running parallel to the vertebral column. There is a lateral channel of the lymph nodes running on the lateral side of the body, and there are lymph nodes near your neck and near the ear and the thoracic area. So these lymph nodes which are there, if you sacrifice one or two lymph nodes, nothing is going to happen to an individual because there are plenty of lymph nodes distributed all over the body. All right. And those lymph nodes, see, here is the diagram that is showing lymphatic channels, lymphatic vessels that are emptying, with large number of lymphatic vessels, afferent lymphatic vessels coming and emptying into the lymph node, and one efferent lymphatic vessel. Okay, so these lymph nodes serve the purpose of expansion of your immune response to the cell mediated immune response, where T lymphocytes will be committed to a given antigen or the antigen that has entered the body, and the B lymphocytes that had come there from the bone marrow have committed themselves to the foreign antigen. Remember that. And when this proliferative activity on encountering the antigen takes place in a particular lymph node, that lymph node will swell. Similar to swelling of the spleen, the lymph node will swell. Sometimes when you have complained of fever, the doctor might have lifted your arm and might have palpitated your armpit and to see whether there is swelling in the lymph node in the armpit. Or sometimes he might have even palpitated your inguinal region, that is the junction between your thigh and abdomen, and palpitated the lymph node there and asked you whether it is paining. So they can palpitate the lymph nodes in different regions. They might have even palpitated the lymph node behind your ear or in the neck region, throat region, by pressing that area. If they feel that the lymph node has swollen there, they can make out that there is some kind of an infection. And then they can draw conclusions like from where all the infections might have taken place, whether it is from the nasal passage or oral passage or GI tract, they can make out. Okay. So as far as the secondary lymphoid organs are concerned, spleen and lymph node are the two major important secondary lymphoid organs. We'll deal with the other secondary lymphoid organs tomorrow. And thank you very much.